This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.
Hey everyone, welcome to Beyond Cancer today. I'm here with my co-hosts Moran and Dr. Mattson, and today we're interviewing Dr. Michelle, and I'll give it right to Brandi to make the introductions.
Great, it's my pleasure to introduce Dr. Michelle Monje. She's a professor of neurology at Stanford School of Medicine and many other affiliated departments as well. So you'll have to ask her about those. But most prominently, she's also an investigator with the Howard Hughes Medical Institute. And on that note, Michelle, can you please share your current and future research activities that you're exploring at the moment?
Yeah, absolutely. So, I'm a basic neuroscientist by training and by practice, and also a practicing neurologist and neuro-oncologist. And my laboratory and my research program for many years has been focused on understanding how the nervous system develops, how activity within the nervous system helps to continually shape nervous system function and plasticity, and how cancers of the nervous system take advantage of both those developmental programs and the activity and plasticity of the brain itself. Did you want more? I can keep talking.
No, I think I'm always amazed about how cancer cells especially love to hijack or co-opt, insert your word of choice, neuronal programs to help to spread and grow.
Yeah.
And I think, as a neuroscientist myself, I think these basic pivotal findings within the field of synaptic plasticities, there is so much that we can use to inform, these cancer neuroscience programs. And to come up with new treatments.
Exactly. The cancers are not inventing anything new. They're simply hijacking and subverting, programs that already exist and physiologies that are already at play in various organs and tissues. And as a neuroscientist, as a pediatric neuro-oncologist, trying to understand how the cancers that I focus on, these really incredibly aggressive, malignant gliomas of childhood, how they arise in very specific parts of the nervous system at very specific ages. There's clearly mechanisms of neurodevelopment that are going awry as these tumors emerge. And so, in the beginning of my career, trying to understand that, trying to understand what cell types were giving rise to these cancers and what developmental events were enabling their growth or encouraging their growth, it seemed really intuitive that the activity of the nervous system itself, which we know to shape all aspects of neural development in the prenatal nervous system, in the postnatal nervous system, to drive plasticity and important tuning and sculpting of the nervous system throughout life, that the cancers might similarly be driven by some of those interactions. And so studying, how glial precursors in the childhood, young adult, and older adult brain are so strongly regulated by activity of the circuits in which they are. And in the healthy brain, that promotes, circuit tuning and in really important ways, plasticity, not just of synapses, but of myelin itself. We wondered whether these glial malignancies might be taking advantage of similar mechanisms. And as soon as we used, techniques of modern neuroscience to ask how activity, within the circuits that the cancer was occurring and might influence cancer pathophysiology, it was clear how very, very powerful a driver nervous system activity was for these primary brain cancers. So
I love how you mentioned the glial cells as well, and the glial biology. And you've been kind of dabbling in that with some collaborations, initially with Ben Barris's lab, as well as his now affiliated trainee, Shane Littleow. So can you explain about what are your thoughts about not just neural activity, but how this broader sense of the neural tumor microenvironment could participate in this process?
Yeah, sure. And perhaps even more than dabbling, I think it's one of the major things that my lab's done for the last couple of decades is to try to understand how neurons and glial cells interact with each other. Ben Barres has always been kind of a scientific hero of mine. I've known him actually since I was a first year medical student. And so his influence on, the questions that I asked as a trainee and as an independent investigator, were he just had a profound influence on my career until, of course, we lost him a few years ago to pancreatic cancer. In studying the way that the nervous system develops and changes in response to experience, one major question in the glial biology field had been, how do myelin-forming glial cells, oligodendroglial glial cells, change in response to activity? There was some really exciting in vitro work and some really, tantalizing correlational experiments that demonstrated that, myelin might change in response to experience. That in addition to the plasticity that we know happens between neurons at synapses, there may be an additional dimension of plasticity that occurs in the glial cells, and in particularly in the myelin-forming glial cells. And by changing in response to experience myelin ultrastructure, one can imagine that there are, sort of fine-tuning of circuit dynamics and therefore of circuit function that might be important for various brain and other nervous system functions. And so this was a big question. There was some controversy about this when I entered this field a dozen years ago or so, because there are also really clearly activity-independent modes of myelination. We know if we put an oligodendrocyte in a petri dish with any, appropriately sized fiber, an inert nanofiber or a formalin-fixed axon, that an oligodendrocyte will ensheath any fiber of appropriate diameter. So there's clearly activity-independent modes of myelination, of myelin development. But we wondered whether in certain circuits, in certain regions of the brain, at certain phases of life or development, whether the activity within a circuit might tune the myelination, that occurs. And so we started using more modern tools of neuroscience to ask those questions, and found that indeed in a circuit-specific and a neuron subtype-specific manner, that glial precursor cells respond to neuronal activity in a very robust way. They proliferate, generate new oligodendrocytes, and also that in addition to de novo myelination of these new oligodendrocytes, that there's also myelin remodeling by existing oligodendrocytes. And so this idea of myelin plasticity, of adaptive and experience-dependent changes in the myelinated infrastructure is something that we and others have been studying now for some time, and have found that these relatively subtle changes in the healthy brain can really contribute importantly to normal cognitive and other neurological functions, while when dysregulated can contribute to a range of different diseases. And so it made sense, given that, glial malignancies like glioblastoma, like the pediatric versions of glial malignancies, including diffuse intrinsic pontine glioma and other diffuse midline gliomas, that they may similarly respond to neuronal activity, in that we think many forms of these glial malignancies arise from precursor cells in the oligodendroglial lineage. And so looking at how, the malignant version of a glial precursor responds to various neuronal subtypes is something that we've focused on now for many years. And we find that just like their normal counterparts, the cancer cells, the glial cancer cells, also respond to neuronal activity and both proliferate, invade, survive in response to the signals that are gleaned.
Oh, that's amazing. I also wanted to ask, in the context of pediatric brain tumors, what are the current challenges in treatment and potential future directions that your research is contributing to?
One of the big challenges in high-grade gliomas and low-grade gliomas of childhood is that in many cases, these brain cancers are incredibly diffusely infiltrative. So rather than being a discrete mass that can be surgically resected, the most common forms of high-grade gliomas in children are intrinsically invasive, and they spread everywhere. There's no discrete mass to resect, and even if there were, they tend to occur in parts of the nervous system, like the brainstem and spinal cord, that we cannot resect. And so there's not really a surgical cure for, the worst forms of glial cancers in kids. And these are one of the most common forms of cancer in children. So this is a very, very big problem. We also find that these are diseases that are not particularly amenable to traditional chemotherapy. So we don't have surgery. We don't have traditional chemotherapeutics. There's been a lot of effort and I think some really exciting progress in the last decade or so to understand the molecular drivers of these cancers and to develop, targeted therapies for them. But in many cases, that effort, as wide and deep as it has been insufficient. And so we are left with a disease that diffusely infiltrates huge swaths of the nervous system and that we cannot surgically resect or fight with chemotherapeutics. And as a result, brain cancer and glial malignancies in particular are the leading cause of cancer-related death in kids. So we have huge challenges. And I think one, point of hope for me is that in understanding the neuroscience of these pediatric brain cancers, there's an entirely new avenue of potential therapeutics that we have at our disposal and can begin to leverage against these diseases. These are cancers that diffusely infiltrate the nervous system. They form synapses with the neural circuits that they're invading. They actually electrically and structurally integrate into the brain. It makes sense that they have been so hard to treat. But now that we understand that, we have a new way to begin to hopefully develop effective therapies.
So, Michelle, in that context, you all nicely demonstrated how those tumors integrate into the existing and de novo, like newly formed circuits. And I've attended a few of your talks and I saw, the beautiful videos that you show in responses of some of your patients to the therapies. I'm curious, the field of cancer neuroscience, even though it's relatively young, there's a lot of data piling both to the CNS and the PNS. And I'm curious, what do you think are the major barriers to take it to the next step, which is a clinical trial? Because I've seen you doing this successfully, in your clinic, in the patient population you're taking care of. And Sean Harvey-Jumper, that also was a guest here in our podcast, was talking about how he aligns his clinical work with his research targeting those newly formed circuits in glioma. And I'm asking, isn't it the right time to move it to clinical trials? And if it is not, what are the barriers? What should we do else?
I think it's the time. And in fact, I've already led one of our discoveries to clinical trial for kids with high-grade glioma. So, I definitely think that, these are therapeutic opportunities to explore and to try to leverage. I think one thing that has emerged over the last, decade or so of the study of the interactions between the nervous system and cancers, not just within the nervous system, but of course outside of the nervous system itself, is that many of the points of interaction, many of the key mechanisms driving these cancers in a neuronal activity dependent way, in a nervous system activity dependent way, are targets we already have drugs for. We have been studying neurotransmitter receptors and ion channels in other disease contexts, and there are really broad range of drugs that have been developed for neurology, for psychiatry, for cardiology, that we may be able to use in an evidence-guided way for cancer. And so that's enormously exciting. And, our job at the bench is to understand for each cancer, for each molecular subtype of that cancer, which are the right, therapeutic targets. And those are tumor specific in many cases. But then, taking the right drug off the shelf and seeing if it can help patients right now while it's simultaneously developing, new and more focused medicines, for disruption of neurocancer interactions are two really important, ways that we can leverage this new information for therapies. And so I think the time is now, as we have the knowledge, to move things from our laboratories into clinical trial. And excitingly, that's already beginning to happen, both in brain cancers, but also more broadly in other cancer types for which a key nervous system mechanism of tumor progression has been identified.
Yeah, well, the reason I'm bringing it up again from the peripheral nervous system perspective, there's a lot of data since Claire Mignon's paper in 2013, the involvement of adrenergic system, for example, and you showed that MDA and GABAergic. So we do have the drugs to target those. We're using those for, like you said, hypertension, or we use those as in epilepsy. And we use those for pain, like for migraine and different types of, but still I haven't seen it, blossoming on the clinical side as nicely as it's blossoming on the preclinical side and on the bench, like you nicely stated. Maybe the time is really now. With that, I would like to transition. We have a lot of your ex-trainees here at MD Anderson, you and Penn, and we already hosted Humsa in the podcast as well. I wanted to ask you about your experience as a mentor in this newly formed field. Cancer neuroscience is kind of a hybrid. It's between cancer biology and neuroscience. What are your major, take-out points or take-home points, for anyone that would like to pursue a career in the field of cancer neuroscience?
Well, the first most important take-home point is it's a super exciting field. So all cancer biologists interested in neuroscience, all neuroscientists interested in cancer biology, all immunologists interested in the intersection of the three fields, please come study with one of us and go in this direction. One of the really exciting, aspects of this field is that it is, as you say, an intersection between disciplines. And I find that often, some of the most exciting progress happens, when there is diversity of perspective, when there's an intersection of fields. And I think cancer neuroscience is a really wonderful example of that. In terms of mentorship, I look for trainees, who come with a background in neuroscience, and the neuroscientist I try to encourage to learn cancer biology. The cancer biologist, I certainly teach, neuroscience. And I think it's really exciting to see this new field emerge at that intersection. But I will say that broadly, mentorship is, IV mentorship is perhaps the most important part of my job. My laboratory has discovered things and will continue to discover things, but that's a fraction of what I hope that our legacy is, which is really to train the next generation of scientists. And so, mentoring is, I think, the most important responsibility and privilege that I have as a professor. And it's my hope that my trainees will, and I know that they will, they already are going out and doing wonderful things. It's really my greatest source of joy.
And you've definitely done a great job, in that. One of your trainees just got the AAAS award, that's Humsa. And we had a great discussion with her here in this podcast. So this is a great opportunity to ask you, what are your expectations from the Cancer Neuroscience 2024 meeting that will be held here at Houston, and how do you see it as an educational opportunity?
Yeah, I think, it's such an exciting meeting. The, opportunity to, as you've done a wonderful job, recruiting speakers who are, from neuroscience, studying cancer, from cancer biology, now studying, neuroscience in that context, immunologists who are, again, studying the intersection of the three fields. And, it's really at these meetings where we bring together the leaders in the field, the thought leaders to, hear each other's ideas and perspectives, that wonderful collaborations can form, that new ideas can spark, and that kind of interaction is invaluable.
That's really amazing. And I know Moran is looking at me, but I also wanted to ask a more specific question. So what inspired you actually to get into cancer neuroscience? Like, what was that defining moment, or did you have a defining moment? I'm just very curious.
Well, I came into the field before it was a field, so okay. As I mentioned, I'm a neuroscientist, and I took care as a medical student of children suffering from diffuse midline gliomas and other high-grade gliomas of childhood, and felt very compelled to better understand it. I noticed the pattern that these are cancers that happen in particular places at particular times. And so I thought that understanding the neurodevelopment of these cancers would be perhaps a new way to look at them and open up potentially a new understanding of how to treat them. So I really just started out with sort of a fundamental search for the biology of these cancers. And as a neuroscientist, one of the most clear questions to ask is, how does activity of the nervous system influence the growth of these cancers? It was remarkable that no one had asked that before in my mind, because we know that nervous system activity regulates every aspect of neurodevelopment. And so as we started to ask those questions and we saw what a incredibly powerful driver of cancer pathophysiology nervous system activity was in these disease contexts, couldn't do anything but study it deeper. It was fascinating. It's important, and I hope that it's going to lead to better therapies for my patients who have such limited options. So have just followed the science. That's really the answer.
That's a great answer. I'm really happy to learn from you, and thank you everyone, honestly, for tuning into our podcast. It's been an amazing pleasure to listen to and learn from Dr. Michelle.
This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.